Experiment 3C. Aim: to measure the water taken in by a plant because of transpiration with the help of Ganong's Photometer. Materials required: Ganong's Photometer, twig of a plant like coleus, knife, water, beaker and coloring agent, cosin. Theory: transpiration is a process in which loss of water in the form of water vapor takes place from the aerial parts of the plant body. It is classified as stomatal transpiration. The loss of water vapor takes place from the stomata, which are minute pores present on the surface of the leaf. These are surrounded with guard cells. The guard cells control the opening and closing of stomata. When the guard cells are turgid, the stomata open, and when they are flaccid, they close. Cuticular transpiration. In some plants, the loss of water vapor takes place from the cuticle present above the epidermis of the leaf or stem. Lenticular transpiration. Some of the old stems of plants have pores called lenticels. When water vapor is lost through the lenticels, it is called lenticular transpiration. Transpiration rate is affected by a number of factors like humidity, light, temperature, wind, carbon dioxide concentration, and water in the soil. These are the external factors. Some of the internal factors are leaf area and structure of the leaf. Significance of transpiration. It influences absorption of water from the soil. The water absorbed by the plant helps in photosynthesis. It helps in cooling the plant. It leads to wilting of the plant. Excessive transpiration leads to stunted growth. Procedure. The twig of a plant is taken. It is cut with a sharp knife and fitted at one end of the capillary tube. The graduated capillary tube is filled with water. One end of the tube is made to dip in a beaker containing colored water. An air bubble is introduced into the horizontal graduated capillary tube by lifting the bent capillary tube above the colored water. As the process of transpiration takes place from the twig, the bubble moves forward. This is because of the suction force which pulls the water from the beaker. Since the capillary tube is graduated, the reading would give an idea about the volume of water lost. Can again be brought into the capillary tube by releasing some water from the stopcock. Observations. As the process of transpiration takes place from the twig, the bubble moves forward. This is because of the suction force which pulls the colored water from the beaker. Since the capillary tube is graduated, the reading would give an idea about the volume of water lost. Conclusion. Transpiration takes place from the leaves and results in absorption of water by the roots. This water then moves up to the leaves through the stem. Precautions. The entire apparatus should be filled with water. There should be no air spaces. The air bubble should be carefully introduced. Limitations of a Ganong's Photometer. A Ganong's Photometer is a device which is used to measure the rate of water uptake by a shoot cut from a plant. It does not measure the transpiration rate directly. The processes of transpiration and absorption are interrelated, and so the rate of absorption is measured and the transpiration rate is assessed. It is a difficult process and requires accurate observation. Hence, it is often not used. In this process, some water is used by the plant for metabolic activities. The rate at which this water is used is not shown by the photometer. The cobalt chloride paper strip experiment is simpler, and it directly helps in comparing the rate of transpiration.